Did your stomach decide it suddenly couldn't take lactose anymore? Well, it's possible you may have undergone a process called DNA methylation. DNA methylation regulates the expression of certain genes by attaching a methyl group to the DNA sequence, essentially turning the gene on or off like a light switch. But how that occurs, I'll discuss later. First, we need to look at this process in action. The agouti gene is a gene in mammals that results in the variation of melanin pigment in the body and can lead to inconsistencies in coat patterns when mutated. The lethal yellow combination, depicted as the dominant big A, little y allele, can result in an abscess of yellow pigment in the fur and other defects like obesity, diabetes, and cancers. A study involving mice carrying the agouti gene demonstrated the effects on supplying pregnant mice with methyl-rich vitamin supplements during pregnancy. Researchers found that the agouti mice who were exposed to the vitamins gave birth to healthy brown offsprings that no longer expressed the agouti gene. The DNA showed that a methyl group had attached itself to the agouti gene and turned it off as a result of the high nutrient diet their mothers had. But why is this important? Agouti mice serve as a crucial model for demonstrating how parental environments can impact DNA in their offspring. While an offspring's underlying DNA sequence can remain unchanged, the susceptibility of which genes are expressed can ultimately be determined by DNA methylation. This process has profound effects on phenotype expression. Triggers for methylation are not limited to methyl-rich vitamins either. Researchers suspect a chemical called bisphenol A, which can be found in plastic drink bottles, can also trigger methylation. Studies show that healthy pregnant mice exposed to the chemical had significantly more obese offspring. Air pollutants have also been associated with DNA methylation, particularly altering the TP53 and EGFR genes, which serve as tumor suppressors that, when altered, can be responsible for the development of lung cancer. While our knowledge about DNA methylation has a long way to come, we do know that methylation leaves biomarkers in our genome that can serve as important indicators for cancer. With this knowledge, early detection and treatment responses have emerged. It's also important to recognize that DNA methylation is a naturally occurring process in our bodies. It controls our gene expression, development, and cell differentiation. Transitions towards obesity and cancers caused by DNA methylation are a result of dysregulation, which is primarily triggered by toxins, poor habits, and pollution, not our usual cellular processes. If you want to learn more about DNA methylation, please check out the source links below down in the description, and I hope you enjoyed my video.